Have you ever walked past a hydrangea and noticed its vibrant blue petals one week, only to see them shift to a delicate pink the next? It's not magic. It's mind-blowing science. Prepare to have your mind blown by what your favorite flowers are really doing behind the scenes. That stunning rose, or vibrant tulip, isn't just sitting there looking pretty. Some flowers are constantly performing incredible chemical feats to change their colors. What if I told you that some of the most beautiful flowers in the world are constantly performing an incredible chemical dance, changing their outfits right before your eyes? This isn't just about pretty petals. It's about a fascinating scientific mystery. We're diving deep into the extraordinary chemical secrets that make some flowers change their colors right before your eyes, unveiling nature's most dazzling transformations. Join us as we uncover the intricate processes that allow certain flowers to dramatically shift their colors, revealing nature's most brilliant secrets. Welcome back to Unmolding Science. The world of flowers is one of spectacular beauty, but the brilliance of certain blooms is not static. It's dynamic. Some flowers are not just one color. They are a living chameleon, continuously transforming their hues in a dazzling display of botanical alchemy. This phenomenal ability to change color, often drastically and visibly, is a sophisticated adaptation, a product of intricate chemistry at play beneath the velvety surface of the petals. Today, we are going to explore the scientific mystery behind the chameleon blooms, dissecting the chemical palette that governs their transformations, and revealing the crucial biological and environmental triggers that motivate these spectacular shifts. The foundation of this color transformation lies in the chemical palette, specifically a group of pigments called anthocyanins. These are the chemical players responsible for the reds, blues, purples, and sometimes even blacks found across the plant kingdom. Anthocyanins are water-soluble pigments found in the vacuoles, the storage sacs of the plant cells. Unlike the green chlorophyll, which handles photosynthesis, anthocyanins are there primarily for coloration, but their appearance is far from stable. The most critical factor governing an anthocyanin's hue is the pH switch, or the measure of cellular acidity. Anthocyanins are highly sensitive to the pH level of the cell sap inside the vacuole. Think of it like litmus paper, a simple science analogy that demonstrates the principle. When the cellular environment is highly acidic, the anthocyanin tends to display a red or pink color. Conversely, when the environment is more alkaline or basic, the very same anthocyanin molecule shifts its structure and displays a blue or even purple color. This explains the classic striking change seen in plants like morning glories, which can open a deep reddish purple in the morning and transition to a true blue later in the day as the acidity in their petals naturally changes with light and temperature fluctuations. This pH-driven transformation is the primary mechanism behind many of the most dramatic color shifts we observe in nature. However, the story is beyond pH. Flower color is rarely a simple one-to-one chemical reaction. Two other sophisticated mechanisms, co-pigmentation and metal-ion influence, add layers of complexity to the botanical palette. Co-pigmentation involves the interaction of anthocyanins with other colorless organic molecules, such as flavonoids or tannins, that are also present in the cell vacuole. These copigments don't produce color themselves, but when they stack or form complexes with the anthocyanin molecules, they stabilize the pigment structure. This stabilization effectively shifts the light absorption and reflection of the anthocyanin, making the resulting color deeper, more vivid, and often pushing the hue toward the blue end of the spectrum without requiring a massive change in pH. It's like adding an optical filter to the pigment. The second crucial factor is the metal-ion influence. This is best illustrated by the iconic color-changing ability of hydrangeas. A hydrangea that grows in acidic soil low pH will typically draw up metal ions, particularly aluminum, from the soil. This aluminum then binds directly to the anthocyanin pigments in the petals. This metallization process is what chemically shifts the pigment from the pinkish hue it would naturally display to a brilliant, stable blue if the same hydrangea is planted in alkaline soil high pH. Aluminum becomes chemically bound to the soil and unavailable to the plant, and the flower reverts to its default pink color. This is a perfect example of a flower using an external environmental factor to literally change its chemical structure and, thus, its color. These chemical changes are not arbitrary. They are driven by environmental triggers and biological benefits, serving a crucial purpose for the flower's survival and reproduction. One of the most common triggers is the change in pollination status. Many flowers change color as they age, or immediately after they have been successfully pollinated. For instance, flowers like the Lantina camara often have a cluster of small blooms where the newly opened, unpollinated flowers are yellow, while the older, already pollinated flowers are red or purple. This shift in color acts as a clear, visual signal to pollinators, such as bees and butterflies. The insect is efficiently directed toward the yellow flowers, which still hold the reward of nectar and pollen, while saving the flower the energy of producing nectar for a bloom that has already done its job. This is a remarkable example of botanical efficiency. Other external factors like sunlight, intensity, and temperature fluctuations can also trigger color shifts by affecting the rate of metabolic processes, which, in turn, subtly alters the vacuole's pH, or the concentration of anthocyanins. The overall biological benefit is clear. By changing color, the flower is fine-tuning its interaction with the environment, optimizing its chances of attracting the correct pollinator at the correct time, or even absorbing heat more efficiently. The scientific understanding of flower color is not just an academic pursuit. It has profound implications for the future of flower color. Plant breeders and geneticists are actively using this knowledge, particularly the role of pH and metal ions, to develop new and exotic flower varieties, attempting to unlock colors that were once genetically impossible, such as true blue roses. By manipulating the genes that control the vacuolar pH or the production of co-pigments, scientists can literally design a flower's color palette. For the viewer, this intricate science should encourage a new level of observation. The next time you walk through a garden or a park, challenge yourself to look for color-changing flowers, perhaps noticing the subtle difference between a young bloom and an old one, or observing how the same species varies its color based on its soil environment. It's a chance to witness botanical alchemy in action, a chemical dance happening right before your eyes. The world of plants is one of remarkable complexity, and the simple act of a flower changing its color is a dazzling testament to the power of chemistry and evolution.